So, and now we go further on the tissue preservation, sequencing facility requirements, and shipment to sequencing facilities. So, so right now what we're going to talk about, actually, we're going to speak about the sequencing requirements and for what sequencing facilities would love you to do, or would love you, you knew before contacting them. So there are tons of different sequencing facilities in Europe. So this is the map of sequencing facilities which are engulfed in European Reference Genome Atlas, ERGA consortium. There are also tons of commercial ones, but these are academic, so they are either based at the research institutes or they are based at universities, and they are non-profit. Some of these facilities, they do DNA extractions and RNA extractions, some do not. At Scilab Lab, we do that. Number of samples and tissues, they might vary. The amount of tissue and DNA that they ask for also varies a lot. Some of them, they accept samples preserved in ethanol. Others will ask you only about for the fresh frozen samples. Also, it's because of the Nagoya and everything else, of course, it's nicer to sequence as close to home as possible. You save also on shipment and also on lots of paperwork. And also one thing that they have in common, though, is that no one can answer how long time it will take and how much is it going to cost. So, and let me try to explain to you what all these things mean. You heard from Henrik, you heard from Yuan, that it would be really nice to get in touch with the sequencing facility also quite early during your genome, or planning your reference genome sequencing experiment. Why is that important? So from the sequencing facility perspective, we could help you with a sanity check of your collection strategies, because we do trust you guys that you know your species best, but maybe you have not done reference genome sequencing before. So we did. So maybe we can help you to avoid some of the hurdles and some drawbacks when you start working on, before actually you go to collect the samples. The sample that you're collecting, it should be good not just to extract DNA, but also to extract quite a good quality DNA. Also, you would like to have RNA, and you would like to have also information for high C. Because if you remember from my first lecture, when you do a classic de novo reference genome, you need to have the long reads, you need to have the chromosome information, chromosome contacts, which is high C. Thomas will cover that in his talk. But you also need to have annotation or RNA-seq to decipher the open reading frames, to understand what proteins are encoded, and so on and so forth. Also, you would like to estimate the required sample size. And based on the organism size, of course, but also based on the genome size, you would need to consider also what is the weight or volume of the sample that you need to bring to extract enough of the nucleic acids. You also need to consider the number of specimens. Is it enough for the one spider or one leaf, or do you need to collect more? Also, the number of tissues for RNA sequencing. As I told you before, the minimal requirement is mixed RNA tissue, but of course it's better to have as many tissues as you potentially can. And also, you would like to calculate the budget. How much is it going to cost? And that budget, if you have some severe limitations, perhaps it will also influence your sampling strategy, and also if you can afford RNA-seq on multiple tissues or not, for instance. So you are out in the field, and Paco introduced you to these dry shippers. And of course, I don't have a mouse. So the dry shippers are God's creation and enormous help for humanity, and us scientists, nevertheless. So they are cheap. They start somewhere 2,000 euros, and you can use them for your lifetime. So your departments might invest in those. So inside, it looks pretty much as a thermos. You saturate that with liquid nitrogen a couple of days before you go to the field. Temperature goes down to minus whatever, 200, and then it can keep this temperature for a couple of weeks. So the dry shippers that I've used, you can use I think after 10 days it still was minus 150. They are safe to transport in the car, unlike liquid nitrogen, and you can also bring them in your carry-on on your flight in all companies but Ryanair. You just need to declare it with your what's the, I lost the word. Button? With a with your air company, your airplane carrier, whatever it's called. Actually, guys, I'm coming down with a cold, so I feel like I'm getting fever. So I'll try not to come too close to you. So if I gave you a hug before, my huge apologies. I felt quite well in the morning, but let's try to persevere. So yes, so the dry shippers, they can be carried on board the plane and train and so on. So that's really good investment. And they freeze your samples directly. And they maintain the very low temperature for quite a long time. So some years ago, we published that in 2022. I was participating in the study from the vertebrate GM project, which is run by the Rockefeller University in collaboration with Sander and some other facilities, and also then with SciLife Lab. And of all the parameters that we have tested for obtaining good quality DNA and high C, temperature was the most crucial parameter. And if you can look over there, different graphs, you see that there are different control temperatures. There is 1 week at 37. There are several months at room temperature. You have 1 week at room temperature, 1 week at 416, and so on and so on and so on. And you can see that the DNA obtained, the fresh frozen, on the first graph, that would be the most to the right. That is the reference. That is that quality of the DNA that we want to get. And if you go to the 37 degrees, which was kept for 1 week, it's already worse. So, but you can also see that you could refrigerate your sample, you collected that in the field, put it in the fridge for at least a couple of hours before you reach the whatever freezer, minus 70 freezer. Even that can salvage your DNA. That works on vertebrates. It doesn't work on insects. Because in case of insects, you have huge contamination of bacteria in samples if you try to preserve them like that. So this data is for vertebrates only. Also, if you look at the yield of the DNA, also of the high molecular weight DNA fraction, and so on and so on and so on, yield per mass, in all of them, you can see that liquid nitrogen or fresh frozen gave absolutely the best result. So absolutely the best DNA quality. Also, if you look at different types of tissues that we use, spleen and blood, we recommend the most from the vertebrates. Those are two tissues that work absolutely the best. With fishes, you can also use gills, but the problem is that there can be some parasites inside. But still, maybe one thing to consider. And absolutely one of the worst is actually muscle. And muscle is something that people usually collect because muscles have lower nuclear density as compared to mitochondria. So you will get lots of mitochondrial data from your muscle tissue. So try to collect spleen or blood. Spleen is actually even better than blood. When you are out in the field and you are collecting your samples, optimally, especially if you do not have access to liquid nitrogen or to dry ice or dry shipper, it would be nice to keep organism alive. It doesn't work with cows, maybe, or maybe yes, but really works really well with some insects. So if this is ethical, it's nice to have it alive as long as you can. If the organism is large, it should be dissected on ice, and you've seen it in Paco's slides. Also, it's a very good idea to remove unwanted tissue, for instance, gut, because otherwise you'll be sequencing the Last Supper instead of the reference organism. However, if you are interested in what this reference organism has eaten, by all means, do sequence gut, but sequence deeper. So you can, bioinformatically, it will be way more challenging to weed out those reads. In case of, there is a new invasive species, at least in Sweden, it's called Abama nungara. That's a worm that eats earthworms. So it's invasive and it's dangerous. And the one that we have sequenced is just ate a worm. And we got 2 genomes for the price of 1 and could not assemble a single one of them because the coverage was too low. So we needed to sequence double just to account for the reads that are coming from the food and not from the insect. And in another case, we sequenced almost the entire spinach genome instead of sequencing snail. So, because the semi-digested spinach apparently is easier to extract DNA from than from the snail. So things like that could really make your life a little bit difficult. Another thing is be very mindful when you freeze. Do not freeze too large pieces because you need the sample to be frozen immediately through the entire volume, especially if you are going to work with RNA later on. So there should be lentil-sized pieces, no larger than that. Don't send us the entire individual because we're on camera. I cannot give you any examples, but I can say that we got whole, say it was, it's from the same genus as, it's from the same taxonomic group as rats. So we got the entire organism in one piece, like solid frozen like that. And for those, we cannot dissect them because of the biosafety legislation. So we had to send it back to the user, and user had to thaw it, dissect it, and of course DNA quality was not optimal, and of course RNA was definitely not optimal. So please do not send us the entire elephant or, any other macroorganism. So if you have a possibility, do weight the sample. It's also good for us to know when we calculate how much buffer do we need for the DNA extraction, if it's us who is going to do DNA extraction. So another thing, it's also from FAQs, the lessons learned. Sometimes we tell users, please give us 10 tubes because we might test different extraction methods. And we've seen tiny spiders who've been separated leg by leg in 10 different tubes. But if your organism is small, please put 1 small organism per tube. Don't try to divide an aphid in 10 tubes. That most likely will not work for anything of what we would like to do. And Alexander, you're welcome to put whole worm in 1 tube. Thank you. Another thing to consider also, place parts into pre-chilled tubes, because otherwise they'll get stuck to the wall of the tube, or it will take a while until the organism will freeze completely. So put the tubes in liquid nitrogen, or treat them on ice, and then put your small tissue fragment somewhere on the pre-chilled tube. And also make sure that the tubes are suitable for -70, so they will not crack. So the Nalgene and Fluidex, the ones that you see in the picture here, they're usually very good tubes to use. So I told you, here is the paper, the reference on the study that we have done. So we tried different types of tissues from different vertebrates. We also tested different types of preservatives. That is in case of absolutely no way you cannot have liquid nitrogen, you cannot have a dry shipper, you do not have ice. What are the other possibilities to avoid the cold chain? Different buffers and solutions that are known to preserve DNA. We tried ethanol, we tried DMSO, and we tried something that is called DNA guard that companies usually promote that will save your DNA if you collect in the field. We also tried with RNA later and see if the tubes sort of saved for DNA or for RNA can be also used for DNA in high C. So why am I mentioning high C so many times? Because for high C, you do not extract DNA. With high C, you start with cells, because optimally you would like to get information from the nuclei. And what high C does, it looks on how DNA is wound around histones, or how is it arranged in the chromosomes. And just to explain to you some terminology, so you see that the red ones, they're coming from chromosome number 1, the blue ones, they're coming from chromosome number 2. We cross-link the DNA to these nucleosomal complexes. Then we chop DNA around, so we preserve only the protein and then small pieces of DNA sticking out. Then we'll remove the protein, ligate with two different ends, and then we make a library, and then we sequence that. So in the picture B, if you see the same color on the same bead, that means this is the close proximity interaction within the same chromosome. That is something that's called cis. And then if you see one red and one blue, most likely they're coming from different chromosomes, and that is called trans. And that is needed to understand the next picture. So this picture says that not all the preservatives are suitable for high C. So the first graph with no preservative, this is a high C of a healthy person, right? When you compare a smoker's lung and a healthy person's lung. So this is a healthy person, high C. Optimally, you would like to have the majority in the dark blue. Dark blue, those are long-range interactions within the same chromosome. That's what you want to have the most of. Gray one is something that is also very important. Those are short proximity interactions within the same chromosome. And still you'll get some of the trans, when chromosomes were close to one another, that can also be informative. So optimally, you would like to have something that's in the picture with blood FF, fresh frozen, and no preservative. When you add ethanol, instead of flash freezing, it's still usable. What you can see is slight reduction in the long cis interactions and more higher fraction of the short cis interactions, right? And look at the DNA guard. It skews the picture completely. You're not getting long interactions at all. And that we have seen with both DNA guard and with RNA later. So that means that if you're going to use sample both for DNA and RNA, or rather divided into different tubes, use RNA later for the RNA and do not add RNA later to the tube, which is supposed to be used for DNA and for high C.
But high C is run on cells, not DNA extractions, right?
No, but you still preserve your sample. Yes, so you still have your cells, your tissue sample, you put it in the preservative, and then we do the mushing and then cross-linking and so on in the downstream library. We do not extract DNA in between. We work with protein wood nucleosomal complexes to start with.
But cross-linking is done with formaldehyde, if I
Yes.
correct. But why can't we just fix the sample in formaldehyde?
Julie, would you like to comment on that?
No, I don't know.
I think, okay, I have an answer to that. So the question was, why can't we preserve our samples directly in formaldehyde? Before we do formaldehyde fixation, we grind DNA in super fine, or not DNA, sorry, the tissue is super fine powder. So it's not, yes, I'll try to explain it then if you come. So it's so fine, it's finer than flour. So that ensures immediate cross-linking of the entire sample in an extremely short period of time. If you dip the entire tissue, I think there will be a gradient of different cross-linking stages through the tissue. So I think that can be one of the reasons. Julie, would you like to comment?
With the cross-linking we have,
I will repeat.
that it's a set time we have it. We don't want to have it like how long is time, like, you know, when have it, but it's set time, then we quench the reaction.
Yes. Okay. So what Tuli said, that Tuli is the lab engineer who is doing high C for the BGA project. So we have a window of opportunity, the specific incubation time when we use the formaldehyde, and then we quench the reaction. That was Tuli's comment. Yes. So basically, in all these graphs, no matter if you have blood or muscle at room temperature or muscle room temperature without preservative, room temperature with ethanol, room temperature with DNA guard, temperature plays a role, so it should be rather fresh frozen, and ethanol should be used as a second best option if nothing else is available. And when it comes to ethanol, the closest to absolute ethanol, the better. So it should be 98, 99%. There are studies when people try to compare different types of methods, and different concentrations of ethanol. And the problem is that very often when people publish such studies, they are concentrating on a certain taxonomic group, something that is close to their heart. So there are no comprehensive studies across different taxa, different parts of the trees of life. But if you are interested for your particular taxa, go online and look for the literature. Maybe somebody done something like that. If you're working with insects, for instance, in the field, and you do not have an access to dry ice, to a dry shipper, you have to use ethanol. Insects and soft shell invertebrates, things like that, put it in the tube with ethanol and squish it gently. So when you squish it, you allow ethanol to penetrate beyond the carapax, beyond the exoskeleton, so it will come to internal organs and to the muscles much faster. And that we have also seen evidence that DNA is preserved so much better when the integrity of the external shell has been compromised. But please do not use DNA guard or RNA later for your high-C samples. So that's exactly what I said. Collect them or dissect in different tubes. If your organism is too small, catch several, put them in different tubes. Because when you generate a reference genome, your DNA, optimally, if you can, your DNA, high-C, and RNA should come from the same individual. But often that is not possible. Annotation is perfectly transferable from different individuals. High-C, it would be nice to have it from the same organism, but not always possible. So again, in some very specific cases, I even seen pooling several individuals together to do high-C, and in some organisms it does work. It works on some very tiny midges, for instance. On nematodes, I think it works as well. So, yes. If you do not have RNA later, trizo is also a very good option. Trizo you can take to the field. In trizo, RNA is pretty stable, also at room temperature for a while, and then it can refrigerate, and it also works quite well. Also, trizo preserves really long mRNA molecules, can also work. And please, if you do not have access to cold chain in the field, try to refrigerate it as fast as you can before putting that in liquid nitrogen. And remember that dry ice or liquid nitrogen are absolutely the best ways to preserve tissue if you want to get good DNA and RNA and high-C out of that. There is a huge treasure trove of knowledge, and it comes from the protocols.io. So for instance, Darwin Tree of Life is constantly uploading new protocols. That's both for collection and for preservation and for different types of DNA extractions. So please go there and have a look if you are unsure. So one can make own DNA extractions, own high-C libraries, and own RNA. But some sequencing facilities are able to provide that. And usually it's done for fee because it takes quite a long time. Also, there are several attempts, different kinds of extractions and optimizations, and so on and so forth. So if we take Sweden, here at SciLife Lab, at Uppsala University here, at NGI, we have set up a lab for high molecular weight DNA extraction only for reference genomes. We cannot do hundreds and hundreds of populations, but we can definitely help you with reference genomes. And high-C libraries, they are done both in Stockholm, and it will be done also in Uppsala. At the moment, we only process BGE samples, but hopefully in September or so, we can open it up for the rest of the country. Also, exactly a year ago, we filmed a digital lab course together with the training hub here. And we are now packaging it, and it's due to be released in August 25. So somewhere in August, if you're interested of making your own high-C libraries and also optimizing your high-C libraries, there will be a full instruction video with instructors wearing GoPros. So you will see exactly the hands, how the things are done, what are the kits, reagents, what do you need to order, and so on and so forth. Also, coming later, there will be courses on demand on extraction of high molecular weight DNA in the NGI Open Lab. More information about that can be found in due time at the NGI homepage. Also, if you don't want, or if you want to extract DNA by yourself, but you perhaps run into some troubles, you're most welcome to contact us at the support@ngisweden.se. We will connect you to the people who are actually doing high-C libraries and DNA extractions. You can have some troubleshooting meetings, perhaps get some tips and tricks. So we are here to help. So use us as a resource. The question, why sequencing facilities ask for many tubes of specimens? So usually our rule of thumb is the following. For doing extractions, we want the more the better. Why is that? If at SciLife Lab and also at other sequencing facilities, we usually deal with the entire tree of life. 1 week it can be 10 crows, another week it can be 1 mollusk and 10 insects, another week can be a plant and a worm or something else. So because of what we're sequencing, what you guys want us to sequence for you, we cannot do comprehensive studies across multiple organism groups, like systematically say that this works for this works for that. So we try to collect all this anecdotal knowledge that we have from different projects, but still sometimes even we fail. We try to do our absolutely best, but in fields of non-model, everything is non-model. We'll try different extraction methods. Sometimes we succeed, sometimes not. RNA extractions usually are the easiest if you preserve the tissue well. So usually for RNA, we need only 1 sample, and if something happens, RNA is difficult, so it's nice to have an extra. And high-C is the same. High-C is even more problematic to troubleshoot and to work with than high molecular weight DNA. So for high-C, there will be also many attempts. And if you want to know more, perhaps you could speak to Tuli at the coffee break. Tuli, would you mind waving? So yeah, Tuli would be one of our main experts here in Uppsala who are working with high-C. And then in Stockholm, we have amazing colleagues who can do that as well. So this is the rule of thumb. For 1 gigabase of genome, it would be nice to have 10 milligrams for animals, fungi, and protists, or 100 milligrams for plants, because of the nuclear density. There are also ultra-low input protocols. We can go as low as a couple of nanograms, but then there is a risk that it will not be very even coverage across the genome. But for some organisms, like unicellular eukarya, that's the only way to go. So then we'll just work with what you have. But if you have multicellular organism, which is large, of course it's better to start with 1, 2, or 3 micrograms of DNA. And if you want to know more, here's the QR code to the Earth Biogenome Project standards that we have updated recently. And the new version is coming anytime now. So this is your checklist that you could take to the field. Think how much material is needed, how many individuals, and in order to know that, you need to contact your sequencing center before you go to the field. Also, what do you need to bring to the field if the sample should be processed there? Can you invest in a dry shipper or a portable fridge, for instance? Because some people, they take portable fridges with them, especially on the cruise, on the big ships and so on. Can your sample be preserved in ethanol? Will sequencing facility that is working with you accept your sample in ethanol? Also, if the genome is supposed to be annotated, bring along the trisol RNA later too for the dedicated samples. And then collect your metadata. You'll hear more from Nicholas after I stop talking. And here, actually, just a thought. It would be nice if somebody developed this fieldwork app, something that you can take on the tablet, only with the internet, you can fill out all these possible fields. I know that in some labs, they're working on it, so perhaps you can check online if something like that is really available. So that would really make your life much more easy. And reminders from Paco's lecture. Make sure the specimen has correct taxonomic ID. Another thing that Paco has not mentioned, aim for heterogametic sex because you want both sex chromosomes, optimally. Take a picture, that's what Paco told you, and deposit a voucher. So if later you would like to go and validate something, people can do that. So this is the sequencing facility documentation, how it works at SciLife Lab. So usually the first point of contact is our order portal. Here's a QR code. Many people of you are familiar in the audience. You sequence genomes with SciLife Lab. You go there, you register your project. After that, a project coordinator will be assigned to you, and that project coordinator will guide you through the entire process, can answer your questions about DNA extractions in some cases. If the person cannot answer, they'll point you to the correct person at the infrastructure. And then the project coordinator will send you an agreement document and send you all the submission forms. And then you sign the agreement, and only then you ship. Why is that necessary? We want to make sure that the samples are traceable. We need to put them in our lab database. If we get a parcel from unknown person with tubes numbered 1 to 25, that will not work. Because at the same week, we can accept hundreds of samples from people all over the world. So that's why it's extremely important to have all the submission information to make sure that we sequence what you want and not something else, to avoid the mix-up of the samples. Shipment. DNA is extremely fragile when it is in solution. So DNA should always be shipped solid, frozen, on dry ice. And here, remember, this is not for PCR. This is for long-read sequencing. So DNA must be solid, frozen. Some courier companies, you heard from Nicholas' lecture, so the dry ice usually does not last more than 48 hours. For many courier companies, you can ask for a top-up with dry ice. That should be asked for separately, and in some cases they say, yeah, we'll do it, but they never do. So, but what you can do your due diligence. You can talk to your courier company and say, we need the dry ice top-up. DHL, FedEx, and UPS, they have destroyed samples in so many different ways. I can write a book about that. We had samples shared by DHL, we have samples lost by FedEx, we have samples which were wrongly declared by UPS, and we still cannot use them because they didn't clear customs in the correct way. So there are two companies I can vouch for. One is the BioCare, it's used to ship human organs. For the transplantation, the clinics. They have all the certificates, and they're really good in legislation. They know everything about Nagoya, they know everything about CITES and so on. Same with World Courier. They are really good. They're not cheap, but it's completely worth it. So why bother about that? All this, why do you want to have really good samples? It's just to give you information here. So sequencing is the cheapest part of your sequencing project. So to generate the data, it's less than 100,000 crowns. But how much time does it, or money it costs to organize an expedition to a different country? How much money do you pay to your students or your bioinformaticians? So when all that adds up, those couple of, dozens of crowns that you pay for the sequencing is really nothing in the big picture. And then also you need to store that data somewhere, so those are costs. Here, don't look at the numbers, they're not, they even are not up to date. They're old, and this is for the Sequel 1 system. But still, I think you should understand the trend. What companies usually tell you, how much sequencing costs. They look at the sequencing reagents, that would be the flow cell or the smart cell or something like that, plus sequencing reagents there, and then the library prep cost. And they do not calculate anything else. We need to do DNA QC, we need to share DNA, we need to make a library, we need to QC the library, we need to make several control checks, and so on and so forth. All that costs extra money. Also, if we look here, that is what sequencing costs if we just take the list prices from the companies. But we also have depreciation agreements for all the possible instrumentation. Also, our runs sometimes fail. So in case if we made a technical mistake, we don't want you to pay extra for that. So that is why we have a distributed cost model when we put 30% overhead on all the costs, so that you do not have to pay for any instrument-related failures. So the price would not be double, it would be only 30% higher. So how long time will it take and how much will it cost? It's really impossible to answer, because non-models, that's exactly what they are. They are non-models, they're recalcitrant. Even some vertebrates, like some fishes, You might think it's easy, but as a matter of fact, it's not. So we need several different kinds of extractions, and also we need to optimize sequencing in some cases. So, and also there is no point in generation of high C if you do not have a reference or the genome assembling, which is of good quality. So all that, it takes time. We need to generate assembly first and we do high C. Also, queues are fluctuating during the year. So it can take everything between 6 weeks and 3-4 months, depending on the complexity of the sample that we're working on. So that is why when somebody is asking how much is it going to cost, difficult to say, we might need to rerun it several times. How long time will it take? Do not know either, because still it can take us several times to make it work. So the take home from this part is, first of all, you can decide what you want to outsource and what you want the sequencing facility to do for you. And get in touch with that sequencing facility early. So we can instruct you about the prices, perhaps help you with a sample, with ideas about sample preservation, and so on and so forth. Do invest time in literature search and check, especially if you haven't done reference genome sequencing before, check in literature in the similar taxonomy groups what people did to achieve their perfect genome. Better safe than sorry, future-proof your samples. So if you were somewhere, collected samples in Chile, Ecuador, or whatever, Australia, or Congo, and you do not have money for your reference genome sequencing, but you want to do it in future, be sure that you're collecting in a way which is future-proofed, that this sample can be used later. And also remember that the sequencing is absolutely the cheapest part of a project. It's your time that costs the most. Yeah? And we can actually go directly to the second part. After that, there will be a lecture from Nicholas about FAIR and the metadata, and then we'll have coffee. Okay? So moving from, you filled out all the paperwork, you came to us, to the sequencing facility, to my colleagues anywhere in the world, and we are doing DNA extraction, you're doing DNA extraction, then we do sequencing, quality control, and protocols.io. So let's start with protocols.io. Protocols.io is an open platform for sharing protocols. It's free. It used to be completely charitable, and now it's under one of the commercial publishing agencies, but still completely open. It's open source, and anybody can go in and free of charge look at everything that is going on. So the parts in the protocol.io that belongs to different groups, organizations, they're called spaces. And these are the 3 spaces that I would like to draw your attention to. This is Darwin Tree of Life space. There is high molecular weight DNA extraction from all kingdoms. That's also a very good one. And then the last one is long read club, especially if you have really weird heterozygous genomes with lots of repeats. You might think about utilization of ultra-low nanopore reads. So these guys, they publish constantly protocols that could help you with that. Soon, I'm looking at Angelo. Angelo, where are you? Angelo decided to leave. But Angelo will come back. So we are trying to create a Silent Lab space and to populate that with BGE protocols. So it's coming. Not yet, but hopefully before the end of the year, we'll have lessons learned from the BGE project. In the field of reference genome sequencing and next generation sequencing, we usually say garbage in, garbage out. Well, we say other words, but for the sake of the recording, let's say it's garbage in, garbage out. Take home here is that NGS quality DNA and PCR quality DNA, they're two completely different kettle of fish, especially for long-read sequencing. And how come? So DNA was discovered at the end of 19th century, and then in 50s, people worked a lot about that. So in good old times, in terms of chagraph, you would go to the closest slaughterhouse, get yourself 54 kilograms of calf thymus, a cow, then you pour like hundreds of liters of methanol on top of that, and lots of salt, and you would extract 1.69 kilograms of DNA. Can you imagine 1.5 kilograms of DNA? Alexander, can you from your nematodes? How many nematodes do you need to sacrifice to get that much DNA? It's many kilograms, right? So then people started also to extract from other organisms, right? So there was the phenylchloroform method by Kirby. And then people started to understand, even with phenylchloroform method, it's not always straightforward. So you need to optimize your DNA extractions if you want to do good molecular biology on that. And then came 83. And 83, they came PCR. And all of a sudden, instead of large molecular biology labs, anybody could do genomics, right? You would go and make your PCR amplicons of your RNA genes and so on, and for that, you would need to extract tons of samples. Who did phenylchloroform extraction before? Yes. How many samples can you do with phenylchloroform? 16 a day? 20 maybe on a really good day? It takes 2 or 3 days, right? So, and imagine doing that for hundreds of samples. So kits came. And kits are nice, cheap and easy, simple, efficient, rapid and expensive. And we forgot molecular biology because now we had kits. And the wake-up call, actually, Mickey in the house. That is one of the first projects we ever sequenced at SciLife Lab. Mickey, this is your DNA. So when we got the long read machines, and at that time we will use like solid and Illumina with short reads and so on. So we got a sample from SLU that was a yeast. And we knew that the genome size is around 7.8. And that's how DNA looked like. And reads were long, so we were very happy. We had some reads above 15 kb. And then we tried to assemble the genome together. So you will hear more from Thomas what these different things are, but N50 of the contigs, that means that the majority of your assembly is in the contigs contiguous sequences of that size, more than 50%, yeah? So it was 2.9 kb. At that time, 2.9 kb in genome assembly, that was really good, right? But not really, but still, quite a bit. But you also see at the sum of the contig length, that is 480 kilobases, and the genome was 7.8. So that data was not enough. We had to sequence it more heavily. And then a couple of months later, we got another DNA, and this team has extracted it differently. And it was a very closely related species. If you look at the graphs on top, you see the top number is 400. In this one, the top number is 7,000. So we got much more reads, also reads which were longer. And look at the assembly stats. We had 9 contigs. That is 8 chromosomes and the mitochondrion. Look at the N50 of the contigs. That's 1.3 megabases. That's the entire chromosome in one contig, in one contiguous gap-free sequence. Max contig length, chromosome size, sum of contigs, 7.8. That's spot on the genome size. How come? So what is the difference between these two samples? So we started digging together with the researchers, and we found out that everything goes back to the DNA. Not just the high molecular weight or not, but also to the chemical purity of the DNA molecules. And here we go back to the point when we have forgotten the molecular biology because now there are kits. Because before there were lots of talks about PCR inhibition. So when you're doing enzymatic reaction, sometimes it simply does not work. With long reads, we do not make any amplification. Long reads, we sequence the native DNA as it is. And from the times when you did PCR or I did PCR, we were sometimes troubleshooting and PCR would fail, right? Why would it fail? Because of the two things. Either there were some chemicals which are DNA binders, or there could be some chemicals which are polymerase inhibitors. These are either carryover from the host organism, from the cells, or it's carryover from our extractions. DNA binders, it's incomplete removal of the proteins, for instance, from the nucleosomes, incomplete removal of polyphenols. Those who work with plants, you know when polyphenol oxidizes, it binds to the DNA, and then DNA will never denaturate. So it just sits there and holds DNA molecules together. They can be secondary metabolites, like in fungi, aflatoxin. It will bind to the major groove and still there, and that DNA is like Gandalf. You shall not pass. The polymerase will not pass through because DNA is unable to unzip. Pigments, polysaccharides, which produce amorphous matrix around the DNA molecule, so enzymes cannot come in. If we are speaking about inhibitors, salts, for instance, EDTA, if you have too much EDTA in your buffers, EDTA is a scavenger of divalent metal ions. So, for instance, magnesium, and what is magnesium? It's a cofactor to the DNA polymerase, so it's food. So if you deprive polymerase of the cofactor that it does need, it's unable to process the DNA further on. If you completely remove phenol, phenol will oxidize your DNA, also introduce single-strand breaks and so on and so forth. Also, when I was a student, I did that as well. Sometimes I didn't want to wait until ethanol evaporates after you wash your DNA. And then, yeah, that's a little bit in there, who cares? So I just add water and try to dissolve DNA. But that's actually bad for your PCR, right? Because that is an inhibitor. So in complete removal of all of that, they can also influence the way how we process the DNA, how we make libraries, and how do we do sequencing and that. So all the living things, but the vertebrates, they have a cell wall, quite a strong and tough cell wall. And what we want to achieve this nice protoplast, and that's from this protoplast we need nuclei, and then we extract the DNA. But those cell walls sometimes are very difficult to breach. So in bacteria, there are different types of polysaccharide complexes on the top of the cell wall. In fungi, there are chitins, the glycopeptides, and so on and so forth. Plants, forget about it, right? Tons of different sugars, tons of different pigments. And in insects, we have the external shell and so on. But we are after the nuclei. So imagine when you extract DNA from whatever the slurry you have after you ground your sample, you want to make a quality control, right? So first of all, what you need to do, you would like to assess the absorption ratios, right? And I'm sorry if I'm preaching to the choir. You definitely know that from your biochemistry, but for the sake of the recording, I'm just reminding that once more. So pure DNA should absorb at 2.8, right? At 260. I'm sorry, I'm getting sick. So it's 260 nanometers, and this ratio should be between 1.8 and 2. If this ratio is lower, that means that we have presence of contaminants that also absorb at 280, which is higher than DNA absorption. It can be proteins, they can be phenols, they can be other kinds of organic contaminants. If it is much higher, then you have something that absorbs at higher than 260. So this is the high share of the RNA. There is also another ratio, 260 to 230. It should be optimally between 2 and 2.2. If it is lower, usually it's salts. They can be peptides, can be some aromatics. Those who extracted DNA from soil, you always get something that is 0.6 or maybe 1.1. Ain't gonna fly, sorry about that. If it is higher than 2.2, still difficult. People do not really know what it really means. Maybe high turbidity, maybe dirty instrument, maybe wrong black, things like that. But the thing is that with this method, we're measuring everything that is photometrically active contaminants, right? Everything that absorbs light. If we're speaking about the kits that we can use still, if we don't want to do phenylchloroform extraction, there is an array of kits on the market, and this is just some small example. There are much more that you can try and see if they work for your organism. So sometimes you have to try different ones in order to succeed. And then you're extracting even with kits or with a C-tub, and you're measuring the absorption ratio. So, oh my God, they're off. Don't despair. There are ways to salvage it. So there are these DNAs, the power cleanup probe, for instance, that works really well, used on soils. and organic matter. And there is also Zymogen DNA cleanup and concentrator kit. Also, one can use ampere beads or any SPRIs, for instance. You can also, after extraction with a kit, you can add an additional layer of cleanup with phenol and chloroform and CETAB if needed, and so on and so forth. Yes, you will lose lots of material at this stage. But think about that. Those molecules that you lose, they were not sequenceable at all to start with. So by purifying your sample and maybe losing some material, still you are salvaging your result. You are salvaging your genomic sequence. This is, again, apologies if you already know that, but that's something that I know that many PhD students are making mistakes. Usually when you take DNA out of the freezer and put it on the table, it falls at the gradient. So if you take 3 different measurements from the top of the well, from the middle, and from the bottom, you have 3 different concentrations. So this is a standard operational procedure that we use at SciLife Lab. We thaw DNA completely. We gently mix, do not vortex. DNA is fragile when in solution. Put it at 37 degrees for a while. You dilute it, especially if it's high molecular weight. And then actually you can make DNA stand on the table for up to 3-4 days. DNA will relax between highly concentrated, high molecular weight DNA. It's very poorly hydrated, right? So the water needs time to penetrate, so DNA will evenly spread. And then you make a measurement with appropriate blank. And then the most controversial part, do not trust nanodrop. Nanodrop is good for one thing, for the spectrophotometric measurement. So from nanodrop, you are getting your ratios to 60 to 230, to 60 to 280. But it measures everything that is photometrically active in your sample. What you want to do, you want to compare the nanodrop measurement with qubit or picogram. This is a different kind of measurement. It's a fluorimetry. So these reagents, or the fluorophores in qubit or the picogram, they'll bind to the hydrophobic part of the DNA, which is existent only if DNA is intact and double-stranded. So, and then you're looking for a ratio between everything that is photoactive in your sample and only double-stranded DNA. So this ratio is less than 50%, most likely your sample is fine. No 100% guarantees, but definitely better than it's more than 50%. So that is something for you to consider. For PacBio sequencing, you do not need to have ultra-long DNA. So it doesn't have to be in 300 kilobases and megabases size. It's perfectly fine if it is 60 or 80 kilobases. But still, the majority of molecules should be quite long. Thomas and I, we published a paper about that and dumped it in bioRxiv because it was like common knowledge review. So all these parameters, how to preserve DNA, and if you want to talk about that, Thomas is here. So go online, look for it, and hopefully it will help you to save your DNA and future-proof it for sequencing later on. Another important thing to keep in mind, there are very many surprises now in the field of non-model genomics. How we think about DNA, it's nice, double helix, right? So the textbooks are telling us that everything is perfect order and so on. Well, this is the model of even distribution of G's and C's on the basis. In the reality, we're discovering more and more tertiary structures of DNA people didn't even know they existed. Through some of them, like G quadruplets, PacBio has enormous problems to read through, especially GA-rich regions. Nanoporting have problems in GC-rich regions. Illumina has problems in GC-rich regions. So the brutal reality is there are things that will not work, and they are dependent not just on the high molecular weight of DNA, not just chemical purity of the DNA, but something biological that can be confirmation bias or something like that. And also the DNA in the solution behaves quite differently as compared to how it does in the living cell. So the workflow, usually I promised you to tell a little bit about the QC, what happens with the samples when they come to us. Either we extracted DNA, you extracted DNA. So first what we do the fluorimetry which is qubit, and compare it either with drop sense or with nanodrop, right? We compare it with spectrophotometry, and we are looking this 1-to-2 ratio, right, or 50% difference. After that, we do something that is called femtopulse. That is a trace up there. What it does, when the DNA molecules are very large, they cannot pass equally nice, let's say, let's simplify it a lot, through the agarose gel. What pulse field does, it changes the orientation of the current and gives DNA more time to squeeze through the pores. Also, the gel that we are using for this particular machine is different from the conventional agarose. So by DNA going up and down, it allows reorientation of the molecules of separation of very long DNA. So on top, it's a good sample, where the majority of fragments are around 170 kilobases in size. So that is optimal. This is something that we would like to do, and then we have a little bit of the tail of fragments down to perhaps 10 kb or so. So that's really good DNA. So after that, we do mechanical sharing. We pass it through the hydropore on the machine called Megaraptor. It's syringe and then DNA by mechanical movement. It shares, so machine shares DNA to the fragment length that we want. Sometimes we ask it, please share to 20, and then we check, and DNA falls apart. That can happen. Or sometimes we tell it, please share to 20, and then give us perhaps 40 or 50. Why is that? Nobody knows, but this is the model genomics for you. So there are lots of surprises and unexpected things. So after we've done that, after we've shared our DNA, we want to again to make sure that the sizes are good. So we do another sizing with a different machine. And then we try to prepare a library. We do the construction, we make several QC steps. Then we do the size selection of the library because we want to get rid of all the shorter fragments that will preferentially sequence. We don't want that. And here is the trace below. My mouse decided not to work today. It took the Valipori one day too early. So, and that is a size selected library over there. That's a different machine, and that library is perfect. It's around 18 or 19 kilobases in size. That's what we would usually like to have. After that, we have the sequencing, and then we do sequencing QC. We look at the number of reads, how well the sample loaded, how fast polymerase was going, and so on. And also something that we do draft assembly and the K-mer plot to estimate if we need to sequence more, or is it enough? So, and after we've done that, we know that we have enough data, then we can give it to NBIS, or we can give it to bioinformaticians for more serious work that Thomas is going to talk about after coffee. And here we have some minutes left, and I'll show you some real-life examples. So that is sample quality versus sequencing. This is a very easy example. Well, people think easy, that's bird. This is a crow, and the genome size is quite small, it's around 1 gig. And that was done a long time ago in a galaxy far, far away in the Sequel-1 instrument. So the PhD student has contacted me in 2015, and he tried to extract from a little bit old sample from a mussel, and it didn't work. You see the absorption ratio is well bad. So he and I, we exchanged 74 emails. So this is the picture how you make a whiteboard pipette tip. 5 o'clock in the evening on Friday, when, I mean, you have to finish your extraction and you cannot wait until the whiteboard tips arrive, right? So you just slice it with a scalpel blade, and that is how you can reduce the strain on your DNA. So finally, 1 month later, just the day before my birthday, he actually gave me a perfect present, so his DNA was perfect. So we were sitting and discussing how to purify his DNA, how to optimize all that. So in the result, we got a beautiful genome. That's, up to now, it's one of the best genomes we ever sequenced, although that was an ownership. That is an example, another example. So on top, that is that particular PhD student and his assembly stats. You see that it's a chromosome scale, it's a very nice size. And here is another person. He was more merited, the researcher, and that was an old sample. We warned that the DNA of that quality will never be the good assembly. The answer was, don't tell me what to do. And the result is, you can compare the assembly stats. So N50 is much lower than 1 megabase, so this will never be a chromosome scale assembly. So I'm not trying to tell you what to do, I'm trying to tell you that perhaps sometimes it's really good to listen to people who've done it a couple of times. So it might help. Plants are tough, so that's another example. We sequenced the maize, and the DNA QC was beautiful. This is pulse field picture. The absorption was spot on, it was long, it was perfect. We start sharing it, it falls apart. Finally, we optimized some steps in sharing with extremely mild. We're making library, we're aiming for 20 KB, we're getting 2. And then again and again and again. So that one took us, from August to February to optimize that. So we did all possible tests. We knew the DNA is chemically pure. So our hypothesis was it's either nicked, because in one of the steps we applied an hexanuclease to remove everything that has not circularized for the PacBio sequencing. Or actually we're thinking maybe there are tertiary DNA structures that can mess up with the sequencing itself. And actually both of that was true. We introduced some DNA repair steps in library construction, and also we looked at the sequence itself. So it were lots of transposons and lots of reads were broken on long transposable elements. So again, some surprises you don't expect. And that particular strain of maize was actually modified in a way that would harbor so many transposons. So that's why we knew we might be up to something. Another example here from EBC actually, so that was an insect that we sequenced quite a while ago. And again, that was a troubleshooting between me and postdoc. We were sitting together again for a couple of months. We tested different protocols, we tested different extraction methods and so on. So finally we got a really nice DNA within the range, chemically pure. We sequenced that and still didn't work. So we optimized again, and finally we tested another kit, and we got, or they got, 2 beautiful papers out of that. We got a really nice chromosome scale assembly that they're using up to now. And this is another example about predictability in inverted commas, of the non-model organisms. So this is our example from SciLife Lab. This is a salamander. So it is exactly the same DNA, the same batch. It was frozen and thawed a couple of times. We did a couple of different smart bells, or the PacBio library attempts, and then we sequenced that. All these variation bars, that's a number of bases that we got from different smart cells. This is, well, you can relate that to DNA quality. That's how many times every read was covered by this hi-fi chemistry. And this is the length of the reads that we got. And you see the distribution, that is exactly the same DNA. And there is no correlation between different libraries, so the variation is between different cells. And the second is actually a more striking example. This is from James Watt, from the Sanger, from the Darwin Tree of Life. He shared this picture with me. So here, the cutoff is what you would say is a successful PacBio run. And in the bars, going like this, these are different taxonomic groups. And in some of them, you can see that you can expect from that much data to this little data. And nobody knows why, because they have standard operational procedures, they optimize their DNA, but some are sequencing well and some do not. And this is from Sanger. They're one of the largest genome producers of non-models in the world. So, before I give floor to Niklas, the take home from here, in the worlds of non-model organisms, everything is non-model. There are lots of different optimization steps either you or the sequencing facility have to do. It works for another species of the same genus. It's not actually 100% it will work. We had examples when we sequenced 5 different bee species. 3 worked like a charm, 2 did not on PacBio. As a last resort, we put these last 2 on nanopore, and it worked. We have another example. Scientists wanted to sequence fish on nanopore. We tried it 5 or 6 different times. It didn't work. We put it on PacBio. It worked from the first time. Ask me why. The answer is I have no idea. It's just, that's unfortunately the reality that we're dealing with. So don't be harsh on yourself. That's not that you do not know how to extract DNA, or it's not the sequencing facility who has no idea what we're doing. It's just the reality. Everybody is facing that. Even Darwin Tree of Life has sequenced more reference genomes than anybody else. And 13 years into the long-read sequencing, it's still a black box. We do not have a single analytical parameter that will tell us a priori if the sample will sequence or not, right? So this is a final note that I would like to shout out to my amazing colleagues who are extracting DNA and doing high-C libraries and trying to optimize the sequencing. So do not forget that that's a huge amount of work that they are putting in. So do not forget to acknowledge them. And if they really spent months and months and months trying to make the samples work, perhaps even more than acknowledging, maybe they even deserve co-authorship. Because we see this shift from molecular biology labs to the outsourcing to the sequencing centers. And now sequencing facility personnel has to become experts on extractions, on sequencing, molecular biology, biochemistry, and so on. So there's lots of work is going into that. All right. So that was it. Thank you very much.